Hi everyone. In this video, we are going to discuss about some of the past paper questions from Unit 7 Human Nutrition, and the paper code is 0610. Okay, so let's begin with that. If you look here, the first question is, the graph shows how rate of an enzyme controlled reaction changes with pH. Okay, so in graph they have given this is rate of enzyme controlled reaction and this one is pH. Now if you look at the graph, this particular line represents the rate of reaction. Now by looking at the graph itself, you can identify the rate of reaction is highest at pH 2. That means the enzyme works best in acidic condition. And as the pH increases, that means as the solution becomes less acidic, the rate of reaction decreases. Okay, so the statements given here are, statement A is the enzyme is destroyed by acidic conditions. Okay, that's wrong. Then the enzyme works best in acidic conditions. The enzyme works best in alkaline condition. Now alkaline pH is starts from 8 to 14. Okay, so no data is available for this third option. And the enzyme best works in neutral condition. Neutral is pH 7, and at pH 7 you can see here the rate of reaction is lowest. Okay, so the correct answer for this question is option B. The enzyme works best in acidic condition, that is at pH 2. Now the second question is, the diagram shows some food moving along the alimentary canal. Okay, this is alimentary canal and the food is moving down as per this arrow. Okay, and I've already explained to people that we have two types of muscles in our alimentary canal, that is circular muscles and longitudinal muscles. So this circular and longitudinal muscles contract and relax alternatively and helps in the movement of bolus of food down the alimentary canal. Now what this process of movement of food down the alimentary canal known as? So the name is, first one is absorption, digestion, peristalsis and swallowing. So this we have already studied and the correct answer is peristalsis. Okay. So the correct answer for question 2 is option C, peristalsis. Now next is question number 3. Question number 3 is, starch is digested by amylase in the mouth, but it is not digested in the stomach. Okay, we know that when we consume food, and in our food if there is any starch present, okay, saliva gets mixed with the food in mouth. Now in saliva, salivary amylase enzyme is present, and amylase is a type of carbohydrates which helps in the digestion of starch into maltose. Okay, now this starch can be digested in the mouth or when the food is passing down through the alimentary canal, I mean through esophagus, but the starch cannot be digested by amylase in stomach. Now you have to tell the reason for this. All starch digestion is completed in the mouth itself. That's wrong. Then the pH in the stomach is not suitable for amylase to work. The starch does not stay in the stomach long enough to be digested. This is also wrong because food stays in stomach almost for 5 to 6 hours. The temperature in the stomach is not suitable for amylase to work. This is also wrong because our internal body temperature in every part of the body is 37 degrees Celsius. Okay, so the correct answer is pH in the stomach is not suitable for the amylase to work. Okay, this is because in stomach gastric juice is secreted, and in gastric juice hydrochloric acid is present. Okay, and this hydrochloric acid is having very low pH, pH 2. So amylase enzyme cannot work at pH 2. Okay, so the correct answer for question number 3 is option B. The pH in the stomach is not suitable for the amylase to work. The next is question number 4. An enzyme from the stomach digests protein, and cooked egg white that contains protein are placed in 4 test tubes. That means from the stomach, in stomach one enzyme is present, and that enzyme actually helps in digestion of protein. Okay, and the same protein which is present in cooked egg. So this enzyme from stomach and the protein which helps and the protein in the form of cooked egg white, okay, are placed in the test tube. 4 test tubes. Okay, now test tube A, B, C and D. Now remember, read the question carefully. What is kept in these test tubes? In these test tubes we have egg white, okay, because egg white contains protein, plus the enzyme which helps in digestion of protein present in the human beings, in the stomach of human beings. Okay, that is there. And we know that the enzyme, protein digesting enzyme in stomach is pepsin. Okay. Now here we have in test tube A, egg white and enzyme is there, and acidic condition. Okay, acid is added. Acidic condition is there, but the temperature is 20 degrees Celsius. In B, egg white, enzyme, acidic condition, and the temperature is 37 degrees Celsius. In C, egg white, enzyme, alkali, temperature is 20 degrees Celsius. Here egg white, enzyme, alkali, and the temperature is 37 degrees Celsius. Now, if you look at the question, it was given in the beginning that enzyme from stomach. Okay, now we know that in human stomach, what is the condition? The condition is acidic. Okay, because hydrochloric juice is released, hydrochloric acid is released in the stomach by the walls of stomach, because pepsin enzyme, which helps in digestion of protein present in stomach, works best only in acidic conditions. Okay, so that means we require acid. Hence option C and option D becomes wrong. Okay, why? Because in this they have added alkali and there is no acid. Now out of A and B, if we'll see, the temperature of A is 20 degrees Celsius and the temperature of B is 37 degrees Celsius. We already know that internal body temperature of human beings is 37 degrees Celsius. Okay, so option B is the correct answer. So in option B, the enzyme reaction will take place faster. Okay, but pepsin enzyme would be most active in test tube B, and hence this test tube will become clear first. So the correct answer for question 4 is option B. Next is question number 5. It's given here. The table shows whether starch was still present after 4 different experiments. This is a similar kind of question like question number 4. In first case, starch plus water at 20 degrees Celsius. Okay. Starch plus amylase at 20 degrees Celsius. Starch plus amylase at 30 degrees Celsius. Okay, and the starch plus boiled amylase at 30 degrees Celsius. Okay, so if you see here, in first case starch was present, obviously. In second case no starch was present. In third also no starch was present, and in fourth case starch was present. In fourth case starch was present as here boiled amylase was taken, and we already know that this boiled amylase, I mean, if we'll if we'll boil any enzyme, if we'll put enzyme under high temperature conditions, then the enzyme gets denatured. Okay, that means the enzyme loses its shape. Okay, loses its structure, hence it cannot digest any starch. Okay, now what broke down the starch? So here the correct answer is amylase, option number A. Next question is question number 6. The graph shows pH changes in mouth after eating. Okay, so after eating, how the pH changes in mouth. Okay, so this is pH, y-axis, and time after eating in minutes. Time is given in minutes. So initially the pH was high. It was alkaline before eating anything because saliva is present in mouth and saliva is 90% water. Okay, so that's why it was actually neutral, and close to neutral, it's alkaline. And after eating slowly, what we are observing as the time passes by, the pH decreases. Okay, that means the acidic conditions increases in the mouth. Now, why it is good idea to brush teeth after eating? Okay, that means why should we brush our teeth after eating? First one is acidic conditions help bacteria to grow. Acid dissolves tooth enamel. Alkaline conditions help bacteria to grow. Alkalis dissolve tooth enamel. Now here these two options C and D gets wrong because there's no alkaline condition or there are no alkalis. Alkalis shown in the graph. Okay, because alkaline conditions start only after 7. Okay, and as you look at the graph, as time passes by, this graph is decreasing down. Okay, if you can see this line, the line is coming down. The graph is decreasing down. Okay. Hence, the correct answer for this question is option B, acid dissolves tooth enamel. Acidic conditions do not help bacteria to grow. Okay, but this acid which is present or which is produced due to the anaerobic respiration of these bacteria present in a mouth, this acid slowly starts dissolving the tooth enamel. Okay, so the correct answer is Option B. Next is question number 7. What is the definition of digestion? Now we already know that in digestion, the large insoluble molecules are changed into smaller soluble molecules. Okay, so D is the correct answer. Large insoluble molecules are changed into smaller soluble molecules. Question number 8. The diagram shows blood vessel P, which carries digested food from the small intestine to the liver. Okay, now this is small intestine, and this blood vessels carries digested nutrients or digested food from small intestine to liver. Now you are already aware this blood vessel P is known as hepatic portal vein. Okay, and it carries all digested nutrients from small intestine to liver for the process of assimilation. Now you have to identify out of A, B, C and D which option best describes the level of glucose and the level of glycogen in liver and blood vessel P respectively. Okay, I mean in blood vessel P what would be the level of glucose, and in liver what would be the level of glycogen. Now first column is glucose in blood vessel P and the second one is glycogen in liver. So option A, glucose in blood vessel P is high. This we already know that it would be high because we have consumed food, the food got digested and these digested nutrients are absorbed into the blood vessel P. Okay, so out of A and B this is correct. Now out of A or B one option is correct. Okay, now after this glucose reaches the liver, okay, the excess of glucose is converted into glycogen. This also we have studied that the excess of glucose is stored as glycogen in the liver, isn't it? So the level of glycogen in liver will increase. So the option B is the correct answer. Glucose in blood vessel P is high and glycogen in liver increases. Okay. Next is question number 9. Which type of tooth is used for crushing food? So the correct answer for this is A. We know that molars and premolars help in digestion of, molars and premolar helps in crushing and grinding of food. So A is the correct answer. C is incisors, B is canine. Okay. Now we have question number 10. This is also based on teeth, but this question is a kind of, it's a key question. Okay. Question given on dichotomous key. So the diagram shows a type of tooth. The upper part of the tooth is known as crown, the part of the tooth which is visible in a mouth, and the lower part which is actually embedded into the gums is known as root. So use the key to identify the tooth. Double rooted tooth or single rooted. So we can see here there is only one single root. Okay. So this is a single rooted tooth. Double rooted tooth are actually present in molars and premolars. Now single rooted tooth we have to go on 3. Flattened 3. Chisel shaped tooth and cone shaped tooth. So this is actually chisel shaped. Okay, this type of shape is known as chisel shaped tooth. Hence it is actually incisors. So option C is the correct answer. Cone shaped tooth means the tooth which has sharp edge like this, cone shaped. Okay, it's sharp like this. Okay, so this cone shaped tooth are actually canines. Okay, so for this question number 10, the correct option is C. Now question number 11. The diagram shows an experiment kept at room temperature. Okay, now this experiment is kept at room temperature. In this experiment you can see there is a test tube. Inside test tube we have a distilled water. Now what is filled? Distilled water, and inside that there is a membrane permeable to small molecules only. This is a semi-permeable membrane, and inside membrane we have starch. Okay, we have protease. Protease is a protein digesting enzyme. We have protein and simple sugars. Okay, now simple sugars are present, starch is present, protease is there, and protein is there. Now what is present in water surrounding the membrane after 45 minutes? After 45 minutes in distilled water, what and all substances will be present? Now we know that simple sugars are, these are the small molecules which can easily pass through semi-permeable membrane. So obviously simple sugar will pass from this tube to the distilled water. Starch is actually a complex. I mean, it's a complex substance. Okay, it's a macromolecule. So obviously starch cannot cross the semi-permeable membrane. So no starch can be found in distilled water after 45 minutes. Protease is the enzyme. Enzyme can also not travel. Okay, but this protease enzyme will digest the protein. Okay, and it will digest protein and convert it into amino acids. So obviously amino acids will also be transferred from this tube to the distilled water. Okay, so the correct answer of this question is question number option A, amino acids and simple sugar. Now we have question number 2, that is a subjective question. You can see here clearly figure is given in which human digestive system is shown. Now you have to use letters on the figure 6.1 to identify. First one is colon. Okay, so what is colon? Colon is actually large intestine, so it is represented by letter P. Okay, the correct answer is letter P. Next is the pancreas. Pancreas is represented by M. So the correct answer is M. And then stomach. Stomach is represented by L. So the correct answer is L. Okay. Let's move on to the next question. Next part of question number 12. Next, they have given state one function for each of the following parts of alimentary canal. Okay. Now this question is given for 3 marks. That means 1 mark belong to each part. First one is colon. Now we already know colon is also known as large intestine, and in large intestine, the food which comes is actually undigested food, and that food is rich in fibers, water, and some of the minerals and vitamins. Okay. So what happens is excess of water gets reabsorbed in the colon. So you can just write down water plus some of the left out minerals and vitamins gets reabsorbed in colon. Next is pancreas. For pancreas, you can write down any function. Okay, you can write down for pancreas like pancreas releases pancreatic juice, which has trypsin, it has lipase, okay, and it also has amylase enzyme, which helps in digestion of different nutrients present in the food. Okay. Apart from this, you can also write down pancreas releases insulin and glucagon hormone. Okay, about this you will learn in further units. And then stomach. In stomach, you can write down pepsin enzyme is present, which helps in digestion of protein into polypeptides. Then next is part C. In part C, question 1 is on figure 6.1, draw a line to show where bile is made. Okay. Now this question is given for 1 mark. If you we know that bile is made in the liver. If you write here answer liver, you will not get any marks. Okay, why? Because they have asked in question to draw a line on figure 6.1 to show where bile is made. Okay, now we know that bile is made in liver. Okay, so you can simply label here liver. Okay, this one. Okay, and you have to label it as X. Okay, so this, and in bracket you can write down liver. Okay, remember you shouldn't label this gallbladder. If you label this gallbladder, you will not get marks. You should actually label liver. Labeling gallbladder is you will not provide you any marks, because in gallbladder bile is actually stored temporarily. Okay, but in liver bile is prepared. Then second, state the action that bile has on fat. State the action that bile has on fat. Okay, in this food. So the food which we consume, in that food if fat is present, so how bile helps in the digestion of fat. So simply you can write down, as this question is for 1 mark, no need of explanation, you can just write down bile helps in emulsification of fat. Okay, if you just write down emulsification of fat, you will get 1 mark. Or you can write down bile helps in mechanical digestion of fat. Okay, that means it helps in breaking down larger globules of fat into smaller globules, so that it increases the surface area for for digestion of fat by lipase enzyme. So here you can just write down bile helps in emulsification of fat, or bile helps in breaking down larger globules of fat into smaller globules. Okay. Next question they have given is explain how this action speeds up the digestion of fat. Now for this you can write down breaking down of larger globules of fat into smaller globules increases the surface area of the fat molecules. Okay. And as surface area increases, enzymes enzyme action speeds up. Okay, so increase in surface area actually helps in faster action of lipase enzyme to break down fat into fatty acids and glycerol. Question number D is, digested food is absorbed as it passes along the small intestine. Now you have to explain how absorption takes place. Okay, how the process of absorption takes place. And this question they have asked for 3 marks. Now we know that absorption of digested food occurs in small intestine. Okay. Now you have to explain how this absorption occurs. Now how you will write, you have to write at least 3 valid points. So first thing you can write down is, small intestine has millions of microscopic finger-like projections called villi. Okay, 1 mark. Then you can write down villi is 1 cell thick. It is provided with blood capillaries, okay, blood vessels and lacteals. So blood vessels helps in absorption of nutrients like glucose, okay, amino acids, some of the vitamins and minerals. And lacteals, they help in absorption of fat, digested fat, that is fatty acids, glycerol, lipids, and the fat soluble vitamins. Okay, so this thing you can write down. Apart from this, you can also write down that these microscopic finger-like projections called villi increases surface area for the absorption of food. So if you write any 3 point out of this, you will get marks. Now the last question in this file is question number 13. Here they have given the enzyme lipase. Okay, they've given here the enzyme lipase digests fat into fatty acids and glycerol. Okay, some students investigated how temperature affects the breakdown of fat in milk using lipase. Okay, so the source of fat here they have used as milk, and we know that in milk lipase is fat is present, and the fat digesting enzyme is lipase. Now here what they are checking, what they are investigating, they are investigating the effect of temperature. Okay, effect of temperature on the action of lipase enzyme. Okay, so here action of lipase is dependent variable and temperature is independent variable. Now they have used here pH indicator called bromothymol. Okay, so this pH indicator was used, and the initial color of this bromothymol was blue. Okay, so this bromothymol was added in all the 3 setups. Okay, and then the color of this bromothymol blue indicator was noted for 2 minutes, for every 2 minutes till the time of 20 minutes. Okay, so for every 2 minutes till the time of 20 minutes, this color of this particular indicator was checked. Now pH and color is given here. At pH 6, the color is yellow. At pH 7, the color is green. And at pH 8, the color is blue. Okay, that means in alkaline condition, the color of this indicator is blue. In neutral condition, the color of this indicator is green. And in acidic condition, the color of this indicator is yellow. Now in cold water, they have taken 3 setups: cold water, warm water and hot water. Okay, this dark gray color test tube. In one test tube they have taken sodium carbonate, milk and indicator. Okay, why they have taken sodium carbonate? Because sodium carbonate is actually an alkali, and we know that lipase works in alkaline condition. It cannot work in a neutral or acidic condition. It works best in alkaline condition. Okay, and they have taken milk because it's a source of food, and they have added indicator. And in next test tube, in one setup, they have taken lipase enzyme in the liquid form. Okay, and they have taken total 3 setups: cold water, warm water and hot water. Now let's look at the other part of the question. Now 3 beakers were labeled cold, warm and hot. 1 test tube was placed in each of the labeled beakers. 3 drops of bromothymol blue indicator were put into each test tube. 2 centimeter cube of sodium carbonate solution was added to each test tube, and then 2 centimeter cube of milk was added to each test tube. Finally, 1 test tube containing 3 centimeter cube of lipase was put into each of the 3 beakers. Okay, and after these 2 test tubes were kept into cold water, warm water and hot water, and the setup was left as such for 5 minutes. And after 5 minutes, what happened? After 5 minutes, the lipase was poured from test tube labeled lipase in the cold beaker into the other test tube in the cold beaker, and the same thing was repeated for the warm beaker and the hot beaker. Now the mixture was stirred and the color of bromothymol blue indicator in each test tube was recorded at start, that means at 0, and for every 2 minutes till 20 minutes. Okay. Now here in this particular picture, they are just actually showing the result recorded by the student. Now if you look here, in first case, in cold water at 0 the temperature was blue, and till 20 minutes at 0, that means at 0 minute the color of indicator was blue, and till 20 minutes the color of indicator was blue only. Okay. At room temperature, that means at 21 degrees Celsius, what happened? The temperature initially was blue, slowly it changed into green. That means, at 12 and 14 minutes it changed into green, and at 16, 18 and 20 the color of the indicator changed to yellow. And in hot water, that means at temperature 50, it was blue initially, then green, and then it has changed to yellow. Okay. Now if we'll compare at room temperature and at hot water, the change of blue color to green was faster in case of hot water than at room temperature. Okay. Now question number 1 which they have given here is, complete the table 1.2 to record the student's result. Now in table they have given time, then color of the indicator, cold, warm and hot. Here you have to mention the temperature. If you will not write temperature here, you won't get marks. Okay. Now where temperature they have given? Temperature is not given initially in the picture, but temperature is given in this particular table. Okay. Ice and water. What is the temperature? 4 degrees Celsius. Now you just have to write here 4. You don't have to actually mention degrees Celsius as it is already written. Okay. So here you have to write down 4, and then warm, that means room temperature was 21, and hot is 50 degrees Celsius. Okay. And then here you have to write down the time 0, 2, 4, 6 and so on. Okay. And then you have to write down the color of indicator. Now here at 0, it is blue. Okay. So you can write down here blue, and then blue. I am writing it as BBB, but you have to write down complete blue. Okay. So you have to make the proper table. Okay. Proper readings. You have to record all the readings in the table, and then only you will be awarded with 3 marks, because this question is for 3 marks. Next question is, sodium carbonate solution has pH of 8. Suggest why sodium carbonate solution was added in the milk. Okay, now we already know that lipase is an enzyme which works best in alkaline condition. So you can simply write down, as this question is for 1 mark, sodium carbonate solution provides optimum pH for the faster or better working of lipase enzyme, as lipase enzymes works best in alkaline condition. Question 2, state why the 2 test tubes in each of the labeled containers were left for 5 minutes before mixing their contents. Now, if you remember, initially they have given the setup of cold water, warm water and hot water. Okay. So in each setup, 2 test tubes were kept. In one test tube, lipase enzyme was there, and in one test tube, bromothymol blue indicator. Okay. Then milk and this thing. Milk and one more thing was there. Okay. Now, why these 2 test tubes were kept separately? Okay, for 5 minutes. This is because both the content present inside both the test tubes should attain equal temperature. Okay, and that attaining of equal temperature will take at least 5 minutes. So content present in both the test tubes should at least attain equal temperature, and after attaining equal temperature only we are going to mix both the the content of both the test tube. Third, explain why the color of bromothymol blue indicator changed during investigation. Okay, this is for 2 marks. The color of bromothymol blue indicator changed during investigation. This you have to explain. Okay, this is because as the time passes by. Okay, as the time was passed by, the action of lipase enzyme was actually working, and lipase was digesting fat into fatty acids and glycerol. Okay, so if you write that lipase helped in digestion of fat into fatty acids and glycerol, and in this fatty acid is produced, hence means acidic condition. Okay, so first the color has changed from blue to green, and then from green to yellow. So for writing this, you will get 2 marks. Then predict the color that you would observe if the experiment had been repeated using water at 80 degrees Celsius. Now the maximum temperature which was which was present in this activity was 50 degrees Celsius. If we'll do the same experiment at 80 degrees Celsius, the color of the indicator would be blue only. Why? Because at higher temperature the enzyme gets denatured. So 1 mark for writing the color of the indicator, that is blue, and 2nd mark for writing that at high temperature the enzyme gets denatured. Hence there won't be any change in the color of the indicator, as enzyme will not digest any fat present in the solution. Now the next question, suggest a result that may be anomalous. Give a reason for your answer. Okay, now you have to actually look at the table again and you have to suggest a reason that suggest a result that is anomalous. The meaning of word anomalous is something like, which is deviating from its own path. Okay, I mean a different result. Now, if you look at the table here, this picture clearly, if you look at temperature 21, okay, room temperature 21, if you see here blue, 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 and at 8 minutes the color was green. And then again at 10 minutes the color was blue, and then green, green, yellow, yellow, yellow. Okay. Now, once the color has changed from blue to green, now the from green again blue color cannot reappear. Okay, that means here there was a mistake in recording the observation. Okay, understood? So here you can just write down there temperature, room temperature 21, and at what particular minutes? At 10 minutes. Okay, and why? What reason you will write? This is because the color of the indicator cannot change as such. Okay, it cannot go back to the previous color. Now question number C, state 2 variables that have been controlled in this investigation. Okay, so for this I'll show you the marking scheme. We actually use dependent and independent variables. Okay, now this question is important. Variable could be enzyme concentration, okay, substrate concentration, the concentration of indicator, and the timing length of the reaction. Okay, so what we can do is first variable, if you're writing enzyme concentration, that means in all the 3 setups, okay, similar or same amount of lipase enzyme should be used. If you will add, for example, in one test tube only 1 ml of lipase, in second 2 ml of lipase, and in third test tube 3 ml of lipase. So obviously the test tube which has more amount of lipase, in that fat will get digested faster, because there actually manpower is more, isn't it? Because as enzymes are more. And next is substrate concentration. That means the fat, the amount of fat. So the amount of fat added in each test tube should be same. Then next was indicator. Same amount or similar volume of indicator should be added in all the 3 test tubes. And then last variable could be timing or length of reaction. Okay, that means minute intervals precisely using timing device. Okay, that means here we have taken the gap of 2 minutes in every reading. Okay, so this also we can change. We can even take 1 minute, or we can take the gap of 5 minutes, or we can take the gap of 3 minutes. But this gap of time, this time gap between each recording of observation should be same. Okay. The last question, last part of this question is, suggest 2 ways to modify this investigation to find the optimum or best temperature for enzyme lipase to break down fats in the milk. Okay. So the correct answer for this question is how you can modify this experiment to find out optimum pH for lipase enzyme. So we can have ideas of more temperature, a bigger range of temperature. Okay, because here we have selected actually cold water, that means around 0 degrees Celsius, and then room temperature 21 degrees Celsius, and then hot water. Okay, that was 50 degrees Celsius. So we can do it as do it at maybe 0, 10, 20, 30, 40, 50, 60 degrees Celsius. And then ideas of the smaller or uniform interval between temperatures. Okay, instead of taking the time gap of 2 minutes, we can choose the smaller gap, 50 seconds or 30 seconds or 1 minute. Okay, time gap of 1 minute between each recorded observation. So by modifying this experiment in this way, by modifying the experiment in this way, will help us in better identification or better finding of the optimum pH for lipase enzyme. So only these questions in this exercise. Okay. In next video, we'll begin with Unit 8, Transport in Clouds. Take care till then. Bye-bye.